Capillary exchange. Exchange means that chemicals are moving in opposite directions. So oxygen and nutrients leave capillaries and go out into the interstitial fluid and from there into the cells of the body. And carbon dioxide and other waste products would leave cells and go into the interstitial fluid and from there into capillaries. So the movement of chemicals into and out of the blood only occurs across capillary walls because they're the only blood vessels that are permeable. And this is where exchange occurs. So most substances will freely diffuse across capillary walls, like water by osmosis, blood gases, glucose, amino acids, mineral ions, vitamins, hormones, etc. are freely diffusible. They move along concentration gradients through capillary walls. Red blood cells and platelets do not cross capillary walls. The white blood cells can move through openings in capillary walls by amoeboid or pseudopodial locomotion, and they do so readily because more white blood cells live outside of blood vessels after they undergo emigration than circulate within blood vessels. So most of the proteins in plasma proteins, do not move across capillary walls. And they're said to be non-diffusible or non-penetrating because they can't penetrate or diffuse through capillary walls. And these include proteins like albumin, fibrinogen, and prothrombin. As a result of these non-diffusible or non-penetrating plasma proteins that can't move through capillary walls and they're retained in the blood, they contribute to the osmotic pressure of blood. So let me just quickly go through this illustration. This would be an arterial, and the arterial feeds blood into a metarterial, and the metarterial delivers blood into a capillary network. Blood flowing through capillaries drains into a thoroughfare channel that carries the blood into a venule. Before we go through capillary exchange, I wanted to talk a little bit about osmosis. So osmosis is the diffusion of a solvent from one solution through a semi-permeable membrane into another solution. The solvent that we'll always talk about when we talk about osmosis will be water. So we'll be looking at the diffusion of water from one solution into another through a semi-permeable membrane. So recall that a solution has two parts. The solvent would be the dissolving medium, always present in a larger amount, and solute would be the chemicals that you dissolve into the solvent. And the solvent and the solute together make up a solution. So if we made a solution of sugar water, we'd get a glass of water, that's the solvent, and dissolve some table sugar or sucrose into it, where the sucrose or table sugar would be the solute that we dissolve into water, the solvent, to make a solution of sugar water. So we'll be talking about the diffusion of water, and I'll abbreviate diffusion, DF. Diffusion is when a substance moves from an area of high concentration to an area of lower concentration. So if we had a substance that was in high concentration, it's said to move down a diffusion gradient, sometimes called a concentration gradient, from an area of high concentration to an area of low concentration. And when a substance moves from an area of high to lower concentration, or down a concentration gradient, it does so by diffusion. A semi-permeable membrane is always a component of osmosis, and a semi-permeable membrane, like the capillary wall, allows some chemicals to pass through it, but not all. It's semi-permeable. I do want you to have a sense of what's meant by free water versus bound water. So free water is water that's not stuck to solute. And free water is the form of water that will diffuse by osmosis through small openings and semi-permeable membranes like a capillary wall. Bound water, on the other hand, refers to water molecules that are stuck or bound by electrostatic attraction to solute that's dissolved in the water. Non-penetrating solute, also known as non-diffusible solute, is solute that is too big to pass through openings in a semi-permeable membrane. So non-penetrating solute consists of mostly the plasma proteins. So it turns out that water will bind to non-penetrating solute, and if the non-penetrating solute can't get through openings in a semi-permeable membrane, then water molecules attached to it wouldn't be able to get through either. So if a plasma protein can't get through a semi-permeable membrane, the bound water can't get through either. The water that is free and not bound to solute like a plasma protein is the water that is able to go through these openings and move from one solution to another by diffusion. And when water moves by diffusion through a semi-permeable membrane, we call that osmosis. Because water molecules will stick to solute, we're going to find that the solute concentration is inversely proportional to the free water concentration. So the higher the solute concentration of a solution, where brackets mean concentration, the more water molecules get stuck to the solute, and that's going to lower the free water concentration. So if you add solute to a solution, some of the water molecules will stick to it, and that would cause a decrease in the free water concentration. The lower the solute concentration of a solution, you'd have fewer water molecules that are bound to the solute, hence a higher concentration of free water molecules that can osmose. So there are a couple terms that are used to describe two solutions that are on either side of a semi-permeable membrane or separated by a semi-permeable membrane. And these are terms that you need to commit to memory. So the two terms are hypotonic and hypertonic. They're coupled terms because if you have a hypotonic solution, you'll always have a hypertonic solution because these are comparative terms. So hypo as a prefix means low, and these prefixes refer to the solute concentration. So a hypotonic solution is the solution with the lower solute concentration and the higher free water concentration. And the hypertonic solution, where hyper means high, is the solution with the higher solute concentration and the lower free water concentration. Now, there are times when both solutions that you're comparing have the same water and solute concentration. If that's the case, then the two solutions are said to be isotonic to one another. So I've just drawn a little illustration to show how you can easily figure out which way water is going to move once you figure out which solution is hypotonic and which is hypertonic. So the hypotonic solution is the one with the low solute concentration and high free water concentration. And it would be separated by a semi-permeable membrane from the hypertonic solution. See that I'm going to see here. The hypertonic solution is the one with the high solute concentration and the low free water concentration. So water is always going to diffuse from an area of high free water concentration through a semi-permeable membrane into a solution with a lower free water concentration. Because osmosis is the diffusion of water from an area of high water concentration to an area of lower water concentration. But we'll always find that water, when it osmoses, will move from the solution with the low solute into the solution with the higher solute concentration. And water will always move from a hypo into a hypertonic solution. So I've drawn a little picture here that kind of shows water osmosing from one solution to another. This would be solution A, and this is going to be solution B. And solution A and B are separated by a semi-permeable membrane. What I've shown as X is going to be non-penetrating or non-diffusible solute. Plasma proteins, for example. So water molecules will stick to solute and form a hydration sphere around them. If the solute, in this case plasma proteins, can't get through these openings because they're too big, then anything stuck to the solute wouldn't be able to get through either. The free water molecules between the hydration spheres around the solute are the water molecules that can move by diffusion through the openings in the semi-permeable membrane. So let's say that solution A is 2% solute and 98% water. And solution B is 10% solute and 90% water. So this solution, solution A, has the higher free water concentration as compared to solution B, and as a result, free water, will osmose through these openings into solution B. And over time, as water diffuses from A to B, solution A will lose some of its water and solution B will gain water. So the solution with the lower solute concentration is the hypotonic solution, and the one with the higher solute concentration is the hypertonic solution. And water will move from an area of high water concentration to an area of lower water concentration. Water will move from the hypotonic into a hypertonic solution. And water will always move from the solution with the lower solute concentration into the hypertonic solution with the higher water concentration. Just a little bit about osmotic pressure. So osmotic pressure is a pressure that you apply to a solution to prevent water from diffusing into it through a semi-permeable membrane. And I've drawn a little illustration to show how the osmotic pressure is calculated. So osmotic pressure, as we'll see, is directly proportional to the solute concentration. We'll find as the solute concentration goes up, the osmotic pressure will go up. As the solute concentration goes down, the osmotic pressure will go down. And we're always talking about non-penetrating or non-diffusible solute, like the plasma proteins. So this will be solution A, and solution A is pure water, nothing but water. There's no solute in it. So 0% solute, 100% water. You cannot get any more hypotonic than solution A. Solution B, on the other hand, is a hypertonic solution relative to A. And let's just say that it's 10% solute and 90% water. It would be hypertonic when compared to solution A. So when you calculate or determine the osmotic pressure, you always calculate the osmotic pressure of a solution containing solute against one that is pure water. So water is going to diffuse from an area of high water concentration through a semi-permeable membrane into one with a lower concentration of free water. And over time, as water diffuses into solution B will gain water. So what you do is you attach a piston to the surface of this water that in turn is associated with a pressure gauge. And as you set up this system, you apply whatever pressure it takes to the water, to this solution, so that water doesn't diffuse into it. So the amount of pressure you apply to this solution to prevent water from osmosing into it is the osmotic pressure of this solution. Capillary exchange involves two processes, filtration and reabsorption. Filtration, which I'll abbreviate with an F, is the movement of water and nutrients in plasma through a capillary wall and into the interstitial fluid. We'll find that during filtration, water and nutrients dissolved in the water of plasma will be filtered out of capillaries and into the interstitial fluid, and this will occur at the arterial end of a capillary. Reabsorption, which I'll abbreviate with an R, is the movement of water in the interstitial fluid through the capillary wall into plasma. So filtration is moving water and dissolved solute out of capillaries. Reabsorption is pulling water back into the capillaries. And reabsorption as a process occurs at the venule end of a capillary. There are two pressures associated with capillary exchange that I want you to know. One of these pressures is blood pressure. Because blood pressure is a fluid pressure, sometimes it's referred to as a hydrostatic pressure. And blood pressure, this hydrostatic or fluid pressure, pushes water out of capillaries, and it's the major force for filtration. I do want you to remember that the blood pressure within capillaries drops from about 35 millimeters of mercury at the arterial end to 17 millimeters of mercury at the venule end. The oncotic pressure. The oncotic pressure is the osmotic pressure due to the presence of non-penetrating plasma proteins in the blood. And the osmotic pressure exerted by plasma proteins in the blood isn't called the osmotic pressure, it's called the oncotic pressure, but it is an osmotic pressure. And the oncotic pressure is somewhere across the length of a capillary, because it doesn't change from the arterial end to the venule end, is somewhere around 25 millimeters of mercury. And we'll find that the oncotic pressure is the major force associated with reabsorption. So the oncotic pressure, in a sense, pulls water from the interstitial fluid into the capillaries. So this is an illustration showing capillary exchange. This would be the arterial, and blood's flowing through an arterial into a capillary, and the capillary is permeable, hence the dashed lines. At the other end of the capillary is a venule that's draining blood out of the capillary. So blood's flowing from an arterial through a permeable capillary, then out the venule. Capillary exchange occurs across the capillary wall. So when you compare the two pressures, blood pressure and oncotic pressure, at the arterial end of a capillary, the blood pressure is greater than the oncotic pressure. The blood pressure is a pressure that's pushing water out, and the oncotic pressure is a pressure that's pulling water in. Because the blood pressure exceeds the oncotic pressure, the net force on the water in blood is to drive it out of capillaries, or there's a net filtration. So 35 for the blood pressure minus 25 for the oncotic pressure equals plus 10. And the plus sign means that water is moving out of capillaries into the interstitial fluid by filtration. Now notice that the blood pressure at the arterial end is 35 millimeters of mercury, but it drops to 17 millimeters of mercury by the venule end, because the pressure of blood declines as blood flows through capillaries. The oncotic pressure, the osmotic pressure exerted by plasma proteins, stays constant. from one end of a capillary to the other and would stay at 25 millimeters of mercury. So if we go down to the venulin, we find that the blood pressure is less than the oncotic pressure. And as a result of the oncotic pressure being greater than blood pressure, there is the net reabsorption of water that's being pulled into the capillary. So if we take a 17 millimeters of mercury for the blood pressure, subtract away the oncotic pressure of 25, we end up with a -8. And the negative sign says that the water in the interstitial fluid is moving into the capillaries. So because the blood pressure exceeds the osmotic pressure at the arterial end, there's a net filtration of water out of capillaries. And because the oncotic pressure is greater than the blood pressure at the venule end, there's a net reabsorption of water back into the capillaries. Now it turns out that more water gets filtered than reabsorbed, and the excess water that gets filtered but not reabsorbed is picked up by lymphatic capillaries and returned by lymph vessels to the plasma of the subclavian veins. Just a little bit about the lymphatic system. So the lymphatic system has several different roles that it plays in the body, and I'll tell you about a couple of those later. But one role is to pick up the water that gets filtered but not reabsorbed, and then to take this water by way of lymph vessels back to plasma. Cholesterol and plasma lipoproteins, new topic. So there are two sources of cholesterol for the body. One source would be animal-based foods, and that provides about 25% of your cholesterol needs. So animal-based foods that contain cholesterol that come into your body with the diet would include eggs, meat, dairy products like milk. In addition to getting cholesterol from animal-based foods, the liver makes cholesterol. In fact, most of the cholesterol, maybe 75%, is synthesized by the liver. If you're a vegetarian and you don't eat any animal-based products, then the liver would make all the cholesterol that the cells of the body need. So why do cells need cholesterol? Well, it turns out that cholesterol is a structural component of the plasma membranes of every single one of the tens of trillions of cells in your body. And in order to maintain these membranes over time, cells continue to need cholesterol. Cholesterol is used by some cells to make steroid hormones like testosterone, estrogen, aldosterone, cortisol. And cholesterol is used by liver cells to make chemicals called bile salts that we'll talk about later. The bile salts help with digestion of fat molecules in the small intestine. When cholesterol moves in the blood, it does so in chemical combination with special proteins called lipoproteins. So we're going to talk a little bit about the transport of cholesterol in the blood and the role that lipoproteins play. So cholesterol and fat molecules, or triglycerides, bind to water-soluble proteins in the blood called lipoproteins. These lipoproteins that cholesterol and fat molecules bind to include three types that I want you to know something about: VLDLs, LDLs, and HDLs. By the way, triglyceride is just a fancy name for a fat molecule. They're one and the same. So let me tell you about each of these three types of lipoproteins that circulate within blood plasma. The VLDLs, where VLDL stands for very low density lipoprotein, are lipoproteins that are made by the liver cells. And the VLDLs include the lipoprotein as well as cholesterol and triglycerides attached to it. The The VLDLs are secreted from liver cells into the blood, and they carry the triglycerides attached to them to adipocytes. Adipocytes would be fat cells. And the fat molecules that adipocytes pick up then are stored as fat in the adipocytes. The VLDLs that lose their triglycerides to adipocytes are then called LDLs. So the liver makes VLDLs, which consist of a lipoprotein, lots of triglycerides, and cholesterol. And the VLDLs circulate in the blood and deliver their triglycerides to adipocytes. When VLDLs lose fat molecules or triglycerides to adipocytes, they are then called LDLs. So the LDLs are derived from VLDLs. And the LDLs contain mostly cholesterol and some residual triglyceride. LDLs. So LDL stands for low density lipoprotein, and LDLs are derived from VLDLs. LDLs carry cholesterol into the cells of the body by a process known as receptor-mediated endocytosis, and the LDLs then give up their cholesterol to the cells once they enter cells, and the cholesterol can be used by the cells for the plasma membranes and to synthesize steroid hormones. So LDLs also can deposit some of their cholesterol and their residual triglycerides into the walls of large arteries, like the coronary arteries of the heart, or the carotids, or the aorta, or the renal artery to the kidneys. Cells often take in more cholesterol from the LDLs than they can use, and the cholesterol in excess of their needs is secreted from these cells into the bloodstream. So there's another type of lipoprotein that I want you to know called an HDL. And HDL stands for high density lipoprotein. The HDLs, like the VLDLs, are made by liver cells, and they're secreted into the blood by liver cells. The HDLs, when they're made and secreted, do not contain any cholesterol or triglyceride fat molecules attached to them. The HDLs circulate within the blood, and they will bind to the excess cholesterol that's secreted by cells. The HDLs can also remove cholesterol from atherosclerotic plaque. So the HDLs take the cholesterol that they pick up as they circulate through the body to the liver, and the liver incorporates the cholesterol into a solution called bile. And bile then, with the cholesterol associated with it, flows out of the liver by ducts into the small intestine. And the cholesterol in bile moves through the intestine and eventually is eliminated from the body in feces. And this is a way that the body gets rid of excess cholesterol. So this is a diagram that takes a look at these three types of lipoproteins. So here's the liver with liver cells, and liver cells make VLDLs and HDLs. The VLDLs that the liver cells make, consist of a lipoprotein, lots of triglycerides attached to it, as well as cholesterol. And the VLDLs that the liver makes are then secreted into the blood and circulate within the blood. Just to reiterate, a VLDL consists of the lipoprotein that's attached to lots of triglycerides and cholesterol. The VLDLs will deliver their triglyceride molecules to adipocytes, where adipocytes would store the triglycerides or fat molecules as fat. And when the VLDLs lose most of their triglycerides to adipocytes, they are then called LDLs. Now, the LDLs consist of lipoprotein, some residual triglycerides, and mostly cholesterol. The LDLs will literally move through the plasma membranes of the cells of the body by receptor-mediated endocytosis and go into cells. And within cells, the LDLs release their cholesterol. And the cells then would use the cholesterol to meet their cholesterol needs. Cholesterol in excess is secreted from these cells into the blood. The LDLs will also contribute the cholesterol that they carry, as well as some of the triglycerides, to the atherosclerotic plaque that can develop in large arteries. Because the LDLs can actually move through the damaged epithelium or endothelium of arteries and deposit the triglyceride and the cholesterol in the wall of the arteries as atherosclerotic plaque. So the HDLs that are made by the liver and secreted into the blood will travel with the blood and bind to this excess cholesterol that's secreted from cells. And it will take that excess cholesterol back to the liver, where it's incorporated into the bile and eventually eliminated from the body in the feces. The HDLs can also strip away cholesterol from atherosclerotic plaque and reduce the size of the plaque.